Hello, and welcome to our MDICx on clinical diagnostic somatic reference samples, our landscape analysis. I'm Carolyn Hiller. I'm the Program Director for Clinical Diagnostics at MDIC's Medical Device Innovation Consortium. I'm delighted to introduce our panelists for today's webinar. Dr. Timothy Stenzel is Director for the Office of In Vitro Diagnostics and Radiological Health, or OIR, at the Center for Devices and Radiological Health at the Food and Drug Administration. Dr. Ivana Tezek is Associate Director for Science and Technology and the Personalized Medicine staff in the Office of the In Vitro Diagnostic Evaluation and Safety, OIR, at the Center for Devices and Radiological Health at the Food and Drug Administration. Dr. Barbara Zensbauer is currently an adjunct professor of pathology at the Emory School of Medicine in Atlanta, Georgia. She's editor-in-chief of the Journal for Molecular Pathology and chair of the steering committee for the SPOT-DX initiative. Dr. Zensbauer has led our efforts on the somatic reference sample landscape analysis. For the webinar, you can enter your questions in the comment box, and we'll have a question and answer session after the presentations that we have. And now I'm just going to very briefly tell you about MDIC. MDIC was the first ever public-private partnership created with the sole objective of advancing medical device regulatory science. We were formed in 2013 to coordinate the development of methods, tools, and resources used in the managing the total product life cycle of a medical device to improve patient access to safe and effective cutting-edge medical technology. MDIC has been designed to pursue several strategies to support its mission. These strategies facilitate stakeholder collaboration to expedite regulatory science innovation and ensure broad-based benefits. Now, what is regulatory science? MDIC's focus is on regulatory science, and simply put, we focus on science, not on policy. Regulatory science is the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of FDA-regulated products. Patients benefit by speeding access to important technologies into their clinical care. Medical device manufacturers and government agencies benefit by reducing time and resources needed for product development, assessment, and review. Whoops, just a second. As you can see, MDIC has a robust program portfolio, and it's focused on a number of initiatives. Our Somatic Reference Samples project is one of our clinical diagnostic projects. Other clinical diagnostics projects include IVD clinical evidence, a surrogate samples harmonized education, IVD real-world evidence, and a fingerstick whole blood capillary collection initiative. You can learn more about our clinical diagnostic programs, as well as MDIC's other projects, by visiting our website, which is www.mdic.org. Again, please enter your questions in the comment section, and we'll have our Q&A at the end of our presentations. And now I'd like to turn the webinar over to Dr. Stenzel.
Thank you, Carolyn. It's a pleasure to be on this webinar. Working with MDIC has really been a pleasure, and will lead and has led to great things already. And this particular effort on NGS samples is so important. Next slide. So in today's world, things have gotten a lot more interesting. Others might say complex. So NGS is more complicated than traditional diagnostic testing because of the enormous amount of data and the high result outputs that result from NGS testing. And we realize at the FDA that we can't use the same old review models when we're operating in this new world. So as technology and knowledge surrounding genetics advances, this traditional model that we've used is no longer adequate to deal with such complexities as we see in next generation sequencing, where one test can provide huge volumes of data, which often cannot be predefined. And sometimes even the disease is not identified until after the test result is received. So this steps away from the old models of just one test, one result, or one test and a few results to looking towards one test with thousands of results. And so as a matter of course, we need to look at ways that we can innovate in this area. Next slide, please. So we're trying to regulate NGS technology-based tests in a way that enables innovation out there in the test development world. And this requires a nimble, inflexible regulatory approach, an approach that can keep pace with the rapid advances in this field. Ultimately, our vision is to accelerate the translation of precision medicine and the technologies related to it into diagnostics and treatments that benefit patients. We are trying to provide an efficient path to market for these technologies while making sure that patients receive accurate and meaningful information. Next slide. So how are we doing this? We have a three-pronged approach. The first part is an approach that details technical standards for NGS-based tests and test development. We envision that these standards would be developed and continue to be developed within the scientific community and can be updated as science, technology, and clinical medicine advance. The second part outlines a process whereby a public database of human genetic variants can be recognized by the FDA, and in fact, we've already recognized one of those databases, and can serve as a source of valid scientific evidence that test developers can use to support the clinical validation of their own tests. And the third part, of which you're going to hear more about today, is to promote the development and characterization of publicly available NGS reference sample sets that can be used to validate NGS platforms. Next slide, please. Now, this is not the only effort in this area. We realize that. We don't want to be duplicative. There's lots of good work out there. And in fact, this list is not even an exhaustive, complete list of the different complementary efforts. Next slide, please. So as an overview to this effort, before introducing the next speaker, I just want to say that the FDA has joined with MDIC to develop cancer genomic somatic reference samples. MDIC is leading this effort, and it includes representatives from academia, pharma, CMS, NIST, FDA, CAP, ATCC, the device industry, and other professional organizations like AACR, AMP, and others. We believe that once developed, these samples can aid in the efficient NGS test development and validation, streamline and possibly obviate steps in the regulatory review process for these tests, provide transparency, and compress development timelines for both devices and therapeutics. Now I would love to hand this over to Giovanna to talk more about this effort. Thank you so much.
Thank you, Tim. So my name is Giovanna Tejuk, and I'm one of the FDA members of the MDIC Somatic Reference Sample Steering Committee. And I'm actually talking today, taking over for J.D. Alvarez, who is the chair of this project, but he couldn't be there today. So I'm kind of using MDIC and his slides. Just want to make the disclaimer that these are not really FDA slides, but I'm talking for the project, not for the FDA. I'm on the steering committee of the project, and I'm representing Office of In Vitro Diagnostics and the Center for Devices. My job today is to give you kind of a short overview of the whole project and really just to set up the main reason for today's webinar, and that's Dr. Barb Zembauer's presentation. she will go into details of the landscape analysis work that she chaired under this project. Next slide, please. Thank you. So as Tim and Carolyn already mentioned, any project under the umbrella of MDIC usually brings together a diverse array of stakeholders. So that's the case with this project too. So the project includes industry, 3 HHS agencies that include payers. So we have CMS, we have FDA, we have CDC at the table. We also have professional societies such as AACR and CAP, academic medical centers, and biorepositories. And you can see them all kind of listed on this slide. What's new to MDIC involvement and contributions for this project is that we have not just device industry, because MDIC is Medical Device Innovation Consortium, right? So most of the projects so far had heavy involvement of device industry, but for this project, we also have pharma industry involved. So that's something different and kind of exciting that we have a bigger umbrella now. Actually, the project chair is J.D. Alvarez, who is from Johnson & Johnson. Next slide, please. Okay, so what is this project? Why is MDIC interested in it? Why are the stakeholders that I was just talking about, why are they interested in pursuing it? This slide kind of gives you the overall goal and the benefits of the work. As already emphasized several times, MDIC projects bring together diverse stakeholders, and they all work together collaboratively towards the same goal. In this case, the main goal is to develop reference samples that can help in efficient NGS test development and validation. And Barb will talk some more about possible other ways to use these materials, and she has a very specific and different expertise. But when we were setting up the goal, and we can see various ways for using this, we can see these samples being useful for a number of other purposes, but the efforts focus and the main goal is to have these materials available for test development and validation. And that goal is of interest to both diagnostic and therapeutic industry and FDA and CMS and other stakeholders, obviously, because having these samples available can be useful to streamline steps in regulatory process, can be useful to provide transparency, and also to compress development timelines for both diagnostic and targeted therapeutic developers. Next slide, please. Okay, so the project is really divided into two main phases. And that is because I think, and Carolyn can correct me if I'm representing this wrong, but most MDIC projects so far have been focused on having the output in the form of some kind of document or white paper, whereas here the main output eventually will be something material, the physical reference samples. So the phase 1 is currently ongoing, and the first product publicly released from this work is this landscape analysis, which is the main focus of today's webinar. Based on the information that's collected during landscape analysis, the work group is also working on identifying the gaps in current somatic reference sample availability, and then selecting which reference samples to prioritize, and working on a plan to develop and characterize and make publicly available these reference samples then. So at the end of phase 1, it's up to MDIC to determine whether they will continue to lead the phase 2 or whether they want to transfer it to the third party to actually make these materials. And then the start of phase 2 will consist of contracting and overseeing a production and characterization of reference samples that are identified as the highest priority during phase 1. Next slide, please. So this slide gives you an idea of who all is involved in the project. At the top, in blue banner, you have listed the steering committee. As I said, it's chaired by J.D. Alvarez from Janssen and directed by MDIC's Carolyn Hiller. You can also see FDI representatives on the steering committee. So we have both representatives for Center for Devices and for Center for Drugs, which again emphasizes the importance of the project for both diagnostic and targeted therapeutic applications. You can also read through and see who all is involved from industry side, professional societies, government agencies, reference sample suppliers, and academic centers. Next slide, please. So the project was officially started last summer, mid-June 2018 at White Oak in Maryland. From the outset, there were 6 working groups that were formed last summer, and you see the first 4 working groups that started working immediately on the left side under phase 1. So that's landscape analysis, variants, sample definition, and business plan working groups. And then under phase 2, you see 2 additional working groups. They were also identified last June, but they are supposed to start their work after the landscape analysis has been completed. So those 2 additional working groups are sample quality and validation and data integration, and both of them are kicking off their work as we speak, considering that landscape analysis has now been published. Next slide, please. Okay, so what are the main questions that this project is set to address? First, what is currently ongoing in this space? Which reference samples are already available? And then which are currently in development, and what efforts are out there? We don't want to obviously duplicate the efforts that others have been doing. There is a lot to be done in this space, and there's no need to replicate. And you've seen in Tim's slide that there are many, many different projects. So we want to know what are the gaps, and then once we identify those gaps, what variants should be prioritized for development, and why those variants? What is the rationale for picking the variants that need to be developed? What sample types are needed? And then the next is how to validate and how to characterize those samples, and how to make those samples then publicly available, and how to make data of their characterization available. One caveat early on, the group has decided to focus on solid tumors and on tissue, so not liquid biopsies, not ctDNA. Next slide, please. In the next few slides, we are sharing with you main description of each of the 6 working groups, but I will not go through those slides because I really want to make time for Dr. Zembauer to go through landscape analysis. So Dr. Zembauer, she's the chair of the landscape analysis. Next slide is sample definition group, chaired by Dr. Mary Ellen DeMars from APCC. Next slide, variant group is co-chaired by Dr. Tim McDaniel from TGen and Eric Peters from Genentech. Next slide is data validation working group that's chaired by Dr. Jason Merker from representing both CAP and from UNC. And then the next slide, data integration, chaired by Lee Tai-Peng from Roche. And the last one, business plan work group that's chaired by J.D. Alvarez from J&J. So this is my last slide, the timeline. This is the timeline for phase 1. So the first year of the project, starting June 2018, ending June 2019. You can see all the working groups color-coded that I've mentioned before, so all 6. and the end of the landscape analysis project is slated for March 2019. So I'm delighted to now introduce Dr. Barb Zembauer, who led that project, to give you the overview and results next. Thank you.
Thank you so much, Giovanna, for that kind introduction, overview, and helping to put this part of the overall SRS project into context. I'm actually here not so much representing Emory University today, but I was pulled into this, recruited, I would say, by Giovanna herself because of my role in another reference sample project for next-gen sequencing, and that is SpotDX, which is included on our report, and you may have heard me speak about that in other venues. Next slide, please. So the aims of the landscape analysis, and these are not just unique to MDIC efforts, there are many ongoing efforts elsewhere of trying to design reference samples for NGS, and everybody's starting out with a landscape analysis. What's already out there? We don't want to duplicate efforts for existing NGS reference samples. That's the first step. We wanted to identify what are the existing genes and variants, as well as sample types that are already out there. But then define what are the unmet needs for additional genes and variants, either for manufacturers of assays or for diagnostic testing laboratories. And importantly, we were just the first step, as Giovanna just outlined. We want to share the data we gleaned from this landscape analysis with the other subgroups of the MDIC SRS group, specifically the sample definition group as well as the variant identification group. Next slide, please. This was definitely a team effort. I'm just speaking on behalf of our subgroup. Listed here are all the members, and they played very active parts in gathering this information, much of it already existing when it comes to commercial products, some of it much more in development for projects that are now in progress, developing other reference samples, so I credit them here. Next slide, please. So what was our process? The members of the subgroup that you just saw, as well as the members of the larger MDIC SRS group, identified NGS reference sample sources as well as ongoing projects, things that they were aware of from their colleagues, from their own work, and that was our starting point. We decided that we wanted to try to collect the following data fields for each of the sources of somatic reference samples. That is basically a project name, a brief description, references, the published reports or web pages that would describe more in detail. Importantly, which genes and variants of those genes, which types of variants were already covered in available reference samples. What types of samples were they? Were they synthetic DNA, genomic DNA, cell-free DNA, human cell lines? Were there any that were formalin-fixed paraffin-embedded tissue that would represent a clinical sample? Then a brief description of what are the validation methods that were used by each source that generated these reference samples, because that would make a big difference to some end users of these materials. Are they commercially available, freely available, and are there contacts with each company or with each project that users may want more information from? And we actively reached out to members of the subgroup to these sources and got more details from them. Next slide, please. For the purposes of our MDIC group, we were just collecting this information in a spreadsheet. And one thing we found early on is that we had some challenges in the information we wanted to gather. First of all was that the types of informed consent or limitations for use of some of these materials is either not available or it's not consistently described. Some of these materials that are available were generated from cell lines established long ago, before we had the current kinds of human subject controls we have now. So we don't always know what the source is or how they're obtained. And that wasn't something we could standardly collect. The other challenge, as many people will recognize, is that many common reference sample types, while easy to produce and perpetuate, may not clearly represent the challenges that laboratories have with clinical specimens in their assay development. Specifically, they may be materials that have already been processed and purified, so that you don't represent the pre-test or pre-analytic variables that clinical samples present to either test developers or diagnostic laboratories. So sample preparation steps may be left out. So you're not really testing for the entire analytic process when you plug in some reference types of samples. Next slide, please. In terms of how do we identify gaps and unmet needs, you know, we racked our brains to identify all the projects we were already aware of, but we wanted to make sure that we weren't leaving out other key resources that were available. So the subgroup members decided to create a short survey of NGS users about what they are using and what their needs are that are still not met. So we developed a very brief online survey. We invited NGS users and assay manufacturers, as you see listed here, to respond to the survey. Next slide, please. The first rule-out question was basically, are you using next-generation sequencing? Because if you weren't, then it wouldn't make sense to respond to the rest of the items in the survey. Next slide, please. So one of the questions we were gathering information about was what types of reference samples were these lab users of NGS currently using. And they could respond to all the types that they were using. So you see there may be multiple responses from each respondent. Most commonly is they're using FFPE, so formalin-fixed paraffin-embedded tissue. That's what commonly presents to diagnostic laboratories in any case for molecular pathology analysis. Cell lines and genomic DNA were also very widely used, as well as synthetic DNA fragments, and this is reflecting what's really available and what's out there. There are limitations I'll talk about in a moment. Next slide, please. We also asked not just what types of reference samples, but what sources, where are you getting these reference samples? A lot of respondents are making use of the commercial manufacturers. Some of them you see listed here. These are not all-inclusive, just examples. And other laboratories report commonly using lab-derived residual samples, leftover patient samples. This is probably one of the most common sources. This is where a lot of those FFPE samples that you saw on the previous slide are represented. The challenges here are that leftover patient samples are not standardized, they're not uniform, even within a block from the same patient or the same tumor sample. They're certainly not standardized across all patients who present with that clinical diagnosis. And importantly, they're not sustainable. They're not renewable. They are very limited. So what do you do when you have to revalidate the assay when that particular control runs out? And there's a few other sources as well. Next slide, please. How do labs use reference materials? If anybody on the webinar has been involved in other reference sample projects, be they SOMATIC or germline, common utility of reference materials is for new assay development, and that's the main goal of MDIC SRS here. New assay development, new validation, streamlining that process by making better defined materials available for everyone to use. The respondents in the survey also stated that they use their reference samples for sensitivity controls, changing the variant allele fraction to represent the different proportions of cancer cells that would be present in a sample. Also importantly, these are run as routine quality controls in lab assays. This is different from certified reference materials that are typically calibrators for testing platforms or instruments. Labs also report using these as process controls. They can be a source of proficiency testing or external quality assessment, sharing specimens among laboratories, that sort of thing. Interlaboratory sample exchange represented here. Next slide, please. For the materials that people were using already, we wanted to get an initial read of what are the important attributes or qualities about the samples you're using that you value. This is important information for the rest of the subgroups in MDIC SRS because it helps inform what are going to be the key components or features of anything we would decide to have manufactured as a result. So which variants are included, that couples with what genes are included, those are very key concerns. How well these reference samples mimic clinical samples, you see is a really high priority and a really key attribute that many respondents cited. Consistent performance, reproducibility, we all know that that's important no matter what your lab setting is. Cost is a concern, although we did not capture cost because that's a changing variable, and that would be something that people would have to be referred to the source material. Availability, what are the range of variant allele fractions that are available? How easy is it to incorporate it into your NGS assay? There are manufacturer available reference samples as well. Will these externally ones be easily adapted into the workflow? And a few other qualities you see listed here. Next slide, please. We also had several different types of comments that we collected from the people that participated in the survey, and you see some of them listed here. We've discussed all of these. The cell-free or circulating tumor cell DNA controls were something that many respondents commented on. As Giovanna said, we decided we cannot do everything in this one project, so that's just deemed out of scope for now. Whether it will be a concern of other MDIC projects, there are other parallel efforts going on. Friends of Cancer Research, for example, Sericare, other groups are working on cell-free DNA controls. One respondent mentioned they really would like to have normal donor plasma. They're trying to mimic some negative controls and make it look more like a clinical sample. Genomic DNA for HPV types, sort of outside of the scope of what we're going to do, but there are some other companion projects that are listed in our report that are addressing that type of application. And then some of these were more comments from labs themselves. Samples containing validated large insertions and deletions are analytically very challenging. There are other groups working on that. I don't know what the final decision will be from our variant subgroup, but that information will be forthcoming. Next slide, please. So how did we create a report? I mean, initially the work of the landscape analysis subgroup was primarily to inform the efforts of the rest of the MDIC SRS subgroup so that they wouldn't be spending time duplicating materials that are already available that people are already satisfied with. So we were just working from an Excel spreadsheet and collecting all of our data. In terms of vetting that information with other people of the project, some folks said, you know, this might be very interesting or useful for labs out there to know all of these different sources and resources all available in one place. So we took the information that we had collected individually and put together, as well as considering the responses that we received during our brief survey, and made that a bit more formal and presentable for public consumption by converting each product or project into its own individual summary table format, which I will show you in a moment. We replicated the key data fields that we had been collecting and made it more into a better description, which you'll see represented. And we added the URLs that would provide links to very comprehensive variant information. Some of these products contain hundreds of variants and many genes, which we didn't want to put into this report and replicate someone else's work. So those links are available for all readers to go to themselves. Pardon me. And then as you'll see, we grouped all these according to their sample type in our table of contents. Next slide, please. This is the current front page of our report as it's posted on the MDIC webpage. You see the URL listed for you here. Next slide, please. And this is just a screenshot of the first page of the table of contents of the report. So you'll see here the organization that we followed, where we have the headings numerically represented of the types of reference samples. So some of the ones that we had are synthetic DNA, genomic DNA, Cell-free DNA is included here because many of the same groups had those, and even though it's not something that MDIC SRS is going to develop, we thought it was a service to the community to put that in there. On the next page, which you don't see here, we also had human cell lines. We have some formalin-fixed, paraffin-embedded either tissue or cell lines, or a formalin-treated DNA because of the insults that that may render to the DNA. So those are part and parcel. We have, I think, a total of 36 different projects or products listed in this report. The next slide contains an example of the standardized summary tables that we created for each one of those headers that you just saw. So this is an example of the page for the American Type Culture Collection product that is the quantitative reference standard. You see on the left-hand column the key data fields that we had collected and their descriptors here. A very brief description of what the product is. We added the URL that links directly to that page that gives you much more details. We briefly described the genes and their key variants that are included in this particular offering of reference sample. As you see, the reference sample type is described. It is quantitative genomic DNA. These all happen to be cell line derived rather than primary clinical samples. The different ways that reference samples are validated is also important for the end user to understand. Most of the information we captured here was available from web pages. They were all then verified with the source of the material that we are representing this accurately, so you see some brief descriptions here. These materials from ATCC are publicly available. Anyone can purchase them. And if you need additional information in the course of figuring out whether this is the right reference sample for your particular application, be it new assay validation, routine quality control, proficiency testing, this is another link that you can go to for additional information. Next slide, please. So vetting the report, the subgroup and J.D. Alvarez, our chair, provided feedback on all of the content information that we provided in those tables in our development phase. In addition, we broke up the work among our subgroup members to go back to the source, back to the companies or back to the project leads for every one of the summaries we had, and verify that the information was accurate, correct, properly attributed. We also found that to be a very beneficial step because in many cases, we had additional projects and materials that were identified by going back to those sources. So we added some more projects to this report. So that was very valuable. Next slide, please. So what's the utility? What is it good for? What do you want to use it for? The landscape analysis report now has already been used by our MDIC SRS project group. They had a long list of genes and variants. They had a public response period for those being posted, what do people need. We then took what they had and cross-checked it with what we were able to identify in our landscape analysis so that we can say if this is already available, we don't need to spend perhaps as much time figuring out whether that is the high priority of what we need to address because we can't make everything. But even for commonly available genes and variants, perhaps the sample types that laboratories and manufacturers are looking for are not adequately represented. So that may be something we'll still keep on our list as a priority. And that's why we chose to break these out by sample type, not just list them all alphabetically, for example. So with additional sample types and variants are still needed is being actively considered by the other subgroups of the MDIC SRS. Next slide, please. The other utility that we projected for this is that laboratories that are performing next generation sequencing, be they assay developers, diagnostic laboratories, researchers, this is really serves as a catalog that summarizes what we know to be currently available and in development, all in one place, all with a standardized format. It's free of commercial endorsement. This is not meant to advocate for using any particular type of sample or source material. It's just all there summarized in an agnostic way with links to more sample details for anyone who wants to know more than what we were able to present. And we also have multiple ways to get this information out to the public that might be users. So one of them is the website. As you've seen on our slides, the full context of the report is currently available as a PDF. You can download it, look at it at your leisure, mark it up, whatever you want to do. This webinar is another way to get that word out, and we welcome all of the people who are listening now. This is being recorded. It will be available for listening at a later time by any of your colleagues who may have missed this particular time. We all have busy schedules. And then also, Giovanna has organized an AACR panel discussion for the end of this month, March 30th at the AACR annual meeting, where the MDIC project and the landscape analysis, as well as a few other projects, will also be described. Next slide, please. And then just as a reminder, here's just a duplicate of that other slide you saw earlier. If you hadn't had a chance to jot down the URL, this is where you can find the report and download it for your own use. Thank you very much for your attention.
Thank you, everybody. And again, you can download the report at the website that we've listed here. And now I'd like to ask our panelists a couple of questions. And the first one I'd like to put out is, why are somatic reference samples important? Again, to touch on that.
This is Barb. I'd be happy to respond to that. So somatic reference samples help us validate the performance of cancer diagnostic tests, and they aid in the development of NGS-based tests for laboratories and manufacturers. These tests are developing very rapidly out there, and it's difficult to keep pace with enough well-designed and well-defined control materials. The FDA has approved quite a few in vitro diagnostic devices, as well as companion diagnostics for personal medicine, but they don't meet all of the clinical diagnostic needs that oncologists have and that clinical diagnostic labs are presented with. So more reference samples for more assay development is something that would be very useful and helpful to practitioners.
Thanks, Barb. Giovanna, Tim, you have anything to add?
I think Barb covered it nicely.
Great. Okay, what can cancer diagnostics professionals learn from the MDIC somatic reference sample landscape analysis?
Okay, this is Barb again. Both the people performing diagnostic assays, as well as the end users of the test results, can learn from the landscape analysis just exactly what is available for, quote unquote, building the quality into your test. Many assays, and if you want to compare performance, or if an oncologist says, how do I know that if I send a patient sample to multiple labs that I'm going to get the same results? Making reference samples available that all users can introduce into their test system can help with comparison of those findings. And knowing that these materials and reference samples are available for any user to purchase or implement into their system is an added level of quality assurance.
And, you know, I think just the document itself is a great reference document for folks that may not know about all the available material out there, and gives them a good place to go to check in case they might have a need. So great effort.
Thank you, Tim.
Thank you. And as a reminder to the listeners, this is an exciting and dynamic space. And so the report in many ways is a snapshot of what we were able to find at this time. There's more coming out almost daily. And so do keep in mind that the reference samples that are currently available may change with time, and the report represents a snapshot of what we were able to find at this time.
Yes, thank you, Carolyn. Yeah, one comment on that. So there are other efforts, as we say, in the field of other groups that are developing reference samples for oncology diagnostics. And what I hear from many groups is the first thing they want to know is, well, we need to do a landscape analysis. What else is out there? So maybe we can help them avoid duplication of effort too by providing this as a reference, a resource for them. And as Carolyn correctly pointed out, this is such a dynamic area of development. We have wrestled back and forth about how can we keep this information fresh and current, and we hope we'll come to some way to do that. So this snapshot may evolve. You could say watch this space, but that hasn't been decided yet. Thank you.
So Carolyn, this is Giovanna. I don't know whether MDIC had some ideas or wanted to collect input maybe from people who are on the webinar, if they have some ideas how you can keep this updated, because as Barb was saying, this is, and as you were saying, this is a snapshot in time right now, and also there are all these efforts ongoing right now that will come to conclusion at some point, and it will need to have to be updated. Because Barb had that one slide where people were, where she was having a survey and asking people what all they want, and there were a lot of ctDNA answers in there, and we know there are several projects ongoing right now for that. So I don't know whether MDIC has some questions or ideas for people out there how this can be kept updated.
Yes, that has been under discussion, and we're grateful for the tremendous effort of all the volunteers that invested quite a bit of time in developing this. In the report on the last page, there is a contact information where individuals are invited to email me if you have questions, comments, and that would certainly be an avenue to alert us of changing information. So I have another question. As we developed this, what was your interaction with the US FDA, and how can this be used by companies to get new NGS diagnostic tests approved?
Giovanna, you want to take that?
Well, I don't think we can answer what was our interaction with US FDA.
But our interaction with test developers has been very good, and you know, in discussing this with members of our review team, they see this as very helpful to test developers. They're often in discussions, you know, they're trying to figure out what samples are available to test these varied mutations. So this effort is very useful for test developers who, you know, want to engage the FDA.
Okay, maybe we can follow up with that. Is what is the challenge of providing somatic reference samples? What are some of the challenges? We've heard a number of them when just by bringing together the diverse stakeholders that all have an interest and are engaged in the somatic reference samples.
So maybe I can start, but I think that we have to tee it off for Barb too. I think somebody mentioned already, this is kind of pre-competitive space. There's a lot of interest in this space, but there's usually no time or no money in this space. So it seems like it's a really good place to bring these all various stakeholders together in kind of pre-competitive space where but we can then use these samples, and I'm now going back to the previous question, we can kind of use these samples for baseline, even in the reviews for starting to evaluate these new tests, whether they're companion diagnostics or whether they're diagnostic, the regular IVDs. It's not end all and be all, of course, for the test evaluation, but it's definitely a good start.
Yeah, I agree with what Giovanna just said. I think part of the challenge is the information and the discoveries and the application of these cancer genetic signatures to diagnosis or treatment selection or monitoring residual disease are evolving so fast. It's very difficult to keep track and find reliable, renewable, affordable, good quality and well-defined somatic reference samples for every test that is involved in oncology diagnosis or treatment decisions. As a result, many commercial manufacturers as well as diagnostic laboratories wind up making their own samples, either contrived samples or surrogate samples when they're validating and developing their tests. But this makes it difficult for some end users of data to compare results across tests or understand how different laboratories perform similarly. So well-characterized reference samples are always going to be continually needed, and therefore some kind of a continuous pipeline would be well accepted by the laboratories that need to incorporate these into their test development or routine quality control. The separate challenge of how to support this financially and make the end product affordable to those groups is sort of beyond my knowledge.
Well, and I think one of the things, this is Carolyn, that from the very beginning as we work on this, the entire effort is focused with all of the stakeholders involved to make sure that the end product, as we identify and eventually create these samples, that they are broadly available, and we want broad access to the researchers and developers who are creating these innovative tests that will help patients with their cancer diagnostics and ultimately selecting appropriate therapies. So when we're looking at access, that could be from a large medical device or pharmaceutical company to a small or an innovative academic researcher in a small university setting. We want everybody to be able to have access to these to advance research for better patient care. Okay, another question that I'll put it out to the team here is, we have this landscape analysis report, and it's now available at no cost for individuals to use. Who are our primary researchers or the users that you would see getting immediate value from using this report?
Barb, you want to take that first?
Sure. Well, I think Carolyn just alluded to a few of those. First of all, people actively working in the laboratories using NGS technology. So researchers, clinical scientists, pharmaceutical companies using these platforms for companion diagnostics development that will target patients who are most likely to respond to their targeted therapies. So people actively in the lab. But the beauty of this project always has been with it's a multi-stakeholder group. So regulators, as Tim and started out speaking about, they're looking for ways to streamline the review process for technology, NGS, that turns out huge amounts of data and really goes beyond the traditional model of reviewing submissions to FDA. And any lab that delivers human samples results, any clinical testing lab or a commercial reference lab, they can use the landscape analysis report to identify sources for quality control reference materials, for their routine quality control assessment, for new test development, and again, for regulatory compliance with their proficiency testing.
Oh, that's excellent. We don't really have anything to add.
Thank you. Okay, are there any closing thoughts that you panelists would like to share with the group as we wrap up our webinar?
Barb, why don't you go first?
All right, thank you. I would just like to say this has just been such an energizing project to be involved in. I mean, I've been working with reference samples for quite some time, but to see everyone coming together from all the different stakeholder groups, not just the laboratories, and saying, yes, we recognize this as a common need. We want to provide our perspectives of what we think is important and necessary and key features to keep in mind. And everybody's a volunteer. They're really working tirelessly in this. And I was just so impressed with the amount of effort and concentration that everyone brought to making sure this information was as comprehensive as we were able to provide and accurate and agreed with what the source of the information was.
And likewise, we feel very energized. We're very excited about being a participant in this effort. We support it very highly and look forward to continued results from this great partnership. Joanna, anything?
And we hope that this will help to, like the goals of the project are to get the great diagnostics and great therapeutics faster to the market.
Great. Okay, everybody, I want to thank you for participating and listening to the webinar, and remind you that a recorded version of this webinar will be available on the MDIC website, and you can find that under the MDICx tab and the archives section. And again, we have the website up there where you can download the landscape analysis. And I want to thank all of you for joining us and wish you a wonderful day. Goodbye.
Thanks.
Thank
Thank you.